Dr. Jackie Ogier. I am a postdoctoral research fellow. I'm an auditory neuroscientist at the University of Melbourne in Parkville, Australia. An auditory neuroscientist is someone who is focused on inner ear biology, so the senses of hearing and balance, and that is the cochlea and the vestibular apparatus in the ear are the sensory organs. And so I'm really interested in the cell types in the ear, how they function, how they develop, how they grow, anything to do with biology in the ear. I started off doing an undergraduate in animal and veterinary biosciences, so I was really focused on zoology and genetics at the time, and that's where I became really interested in biology. I didn't know that that's where my career was going to go, so I actually finished my degree and ended up working as a tech in a research facility. I just needed a job at the time to pay the bills. But in that role, I actually came in contact with researchers daily. And there was one researcher in particular who would often speak to me about her work, and I found it really interesting. And she said to me, look, you ask the right questions, you're obviously really interested, would you consider doing honours in my lab? And I actually said no at the time. I thought I was done with university, I didn't want to go back. But she kept asking, thankfully. And at the time I started to realise that my job felt a bit stagnant, so did my life. And I thought, this is an opportunity that I would be silly not to take. It's a leap into the unknown, and it was a really defining moment for me because I ended up saying yes. I ended up doing an honours year in the molecular hearing laboratory at the Murdoch Children's Research Institute, again in Parkville in Victoria, Australia. And during this time, I really fell in love with the process of research. So I found out I was quite good at it. I really enjoyed the creativity of having a question that no one knows the answer to. You can't find it in a book. You have to design an experiment that is controlled and really hones in on answering that question. And it was very exciting to me. So I was quite hooked. And in that role, I was investigating the genetics of hearing loss. So trying to understand and identify new genes that could potentially contribute to hearing loss in the population. And that was also my first introduction into hearing and balance as senses. I think we take for granted how complex that whole system is. And so after my honours year, I stayed in that lab and I worked for a couple of years until I started to feel that I wanted to take more control of my research journey and have my own independent research projects. And so that's when I pursued my PhD. And so for my PhD, I actually moved even more into cell biology, where I started to specialise in the sensory hair cells in the ear. Now, they're called hair cells, but they've got nothing to do with the hair that grows on your head. They're named more over their shape. They've got quite a long shape with stereocilia on top that look a bit like hairs. But these cells are the interface between the outside world and our central nervous system. So they've got this really cool job where they take what is a mechanical stimulus and they convert it into an electrochemical signal. So this process, which is called mechanotransduction, if you think about it in the context of sound, sound is a mechanical vibration that is moving through the air. And these cells, when they're stimulated by this vibration, they open mechanically gated channels that then causes an electrochemical change in the cell, and ultimately neurotransmitter is released at the base of the cell, causing an action potential to send a signal to the brain. And the same cells do a very similar thing for balance, but they, instead of detecting a vibration, are detecting fluid movements caused as you, say, move your head or move backwards and forwards. And so they're really important for you to understand where you are in the world and your sense of self. So during my PhD, I was really interested in how these cells die, because you're born with a certain number, and over the course of your life, they get damaged and you lose your hearing and your balance as a result of that. And so for my PhD, I was investigating a new molecular target that we could potentially target to prevent hair cell death, particularly in the context of ototoxic medicines, which are life-saving medicines that can have a side effect where they damage hearing or balance. And I was able to identify a novel drug that we developed some preclinical evidence for to show that when we used this new inhibitor, we could prevent hair cells from dying in a dish when we used some of these ototoxic medicines. So that was a really exciting finding for me and my first real foray into the cellular bioscience level. As I came to the end of my PhD, I was offered a role in Canada, a postdoctoral role. Unfortunately, the pandemic hit, so it was a little bit interrupted, but I did manage to head over there and did some really exciting work where the team in the Dubdoob lab at the Sunnybrook Research Institute in Toronto were getting donated human ears. So this was either from people who had donated their ears after they had died, or during surgery, if people were having maybe a tumour removed from the ear, sometimes we would receive a sample that way. And this research was really exciting because we were using single cell technology to better understand all of the different cell types in the ear. And this was a moment that really helped me to see that inner ear biology and our research, we do lag a little bit behind because it's impossible to biopsy the ear. So most of us think of the ear as this external component, but the part of the ear where the sensation happens is very deep within your skull. So it can't be biopsied, it can't be easily collected, we can't get tissue samples from it. So we really rely on these very rare donated samples. And that's where I started thinking about inner ear organoids as an approach to expand the scope of our research. And I was lucky enough to get support from the Passe and Williams Foundation. They provided a fellowship, which meant I could come back to Australia. Now I'm working at the University of Melbourne in the Nayagam lab, and I'm working in the Centre for Stem Cell Systems. And what we do is we use human-induced pluripotent stem cells. So these are cells that we can just start off with a skin sample. We process the cells to make them into a pluripotent cell that behaves more like an embryonic cell. And we can treat them in a dish with various factors that replicate development. And what we end up with is we grow these organoids, which are basically a big sphere of cells, and they end up differentiating into the cell types that we find in the ear. So there's cells that resemble the sensory hair cells that I've spoken about. There's also neurons, there's supporting cell types. There's even evidence that we have the calcium crystals that are part of the balance system. And so this is a really exciting way that we can grow the cells that we want to investigate in a dish. So it's important because it means we can move away from animal research, and it also means we can really expand what we're doing. So it's a scalable tool. And we can genetically alter them so that we can have certain cell types that fluoresce a colour. That means we can focus very easily on that cell type. And using organoids, we can investigate, like I did during my PhD, different drugs that we could potentially use to prevent hearing loss or balance loss. We can also really try to home in on how these cells develop, and that could have really big implications in the future for regenerative medicine and also potentially personalised medicine. So if we go back a step and we take cells from a person's skin and we start the process, that means that if they have a particular type of hearing loss, we could potentially tailor drug treatments to that person. Obviously, this is far in the future, some of what I've just said, but it's a really good example of how organoids can really expand the scope of what we're doing at the moment and supplement those donated samples that we're getting. I'm also lucky enough to be able to continue working on some of what I did during my PhD and build on that. So I'm doing a proteomics project where we're really trying to map all of the proteins that are in a sensory hair cell, and then we can also treat sensory hair cells with ototoxic drugs and then do that same process. And working with the amazing bioinformaticians at Bio21 at the proteomics facility there, we can then map out the changes in signalling that occurs. And so using proteomics, we're hoping to get a really high resolution map of what is happening in a dying hair cell, and that will help us to identify new drug targets, potentially compensatory mechanisms that we can target to improve hearing retention in people that are experiencing something that might cause hearing loss. And I'm also working with some amazing engineers at the Graham Clark Institute, where we are designing a new coating for cochlear implants. And the idea with this new coating is that it will make the implant last longer and be more biologically safe in the ear, and hopefully that means that people who receive the cochlear implant will have better speech outcomes in their the duration of their life, because we get cochlear implants quite early in life sometimes, and they stay in for the rest of our life. And so by having this new coating, we really hope to improve human health that way. I'm passionate about hearing loss in particular. My family is 3 generations have had early onset hearing loss, so there is that personal connection. But also just for me, I think hearing, we take for granted that it is a sense that is so integral to the human experience. So you're always hearing, and it's never really switched off. And we also know that as you lose your hearing, there's some really significant impacts on your brain health. So this might start as mental fatigue, when as hearing becomes more difficult, you have to put more effort into listening. And with that effort is a higher cognitive load. And we know from plenty of research that's been done that when you have that cognitive load focused on hearing, it becomes really difficult to make decisions. Remembering things becomes more difficult, and just you can experience exhaustion. Over time, this can become even worse, where the more hearing loss you have, you start to have structural changes in the brain. You might also withdraw from social situations because it's a little bit too hard. And overall, this actually contributes to a much faster rate of cognitive decline. So we know as we age, if you have untreated hearing loss, you're far more likely to develop Alzheimer's disease, and also just have more difficulty regulating emotions and understanding what's happening around you. But the good news is that this is for untreated hearing loss. So something as simple as a hearing aid can completely reverse this trend. And I can speak from personal experience with my family that a young member of my family was saying that before they got hearing aids, they were going to work, working as a teacher, and coming home so exhausted that they had to go to bed before they could even think about making dinner. And they said, I've just got a very demanding job. And now that they've got 2 hearing aids, they come home and they've got plenty of energy to get through the rest of the day. And having her say that to me was really quite enlightening, because I think we really do take for granted how easy listening is when your hearing is good. And we really don't notice it as it's going, because it tends to go quite slowly. And so most people don't seek help until they've been living with hearing loss for sometimes decades. And the reality is 1 in 5 Australians actually has significant hearing loss. So if you take one thing home from this, I really hope you consider getting your hearing checked. You know, if you woke up and suddenly everything was blurry, you would get your eyes checked. So consider getting your hearing checked, because the sooner you do, the sooner you can get an intervention that will just make life easier. And so going back to my research, I really hope that to add to the support devices that we have, so cochlear implants and hearing aids, hopefully I can add some biological interventions that could potentially either improve hearing or at the very least prevent hearing loss from happening in the first place. Outside of work, I've got a number of hobbies. Music is a really big one for me, so that might also be a reason that I'm really interested in hearing. But for me, as a scientist, it can be really difficult to switch my brain off at the end of the day. It's hard not to think about the experiment that didn't work, or maybe a new question you've thought of and how you're going to answer it. And so being in a choir, so I'm in the Melbourne University Choral Society, and I'm also in the Essendon Symphony Orchestra, which is a community orchestra. I find that the process of sight reading music while listening and engaging with the people around me, while also engaging my body to make that music, it's a great way of just switching off. So even though I'm very active in that process, it really helps me to just take a break from research, because I think most researchers find that it's really hard to switch off. And also through those experiences, I've had other amazing experiences. So with the choir, we recently performed with the Royal Melbourne Philharmonic at the Town Hall, doing huge symphonic works. We've toured Europe, and I've seen places that I never would have seen if I didn't participate in that. And then aside from music, I am also a beekeeper. And so I find bees, their social structure, their biology is really interesting. And so I love getting outside and working with bees. I often am called to rescue a swarm or to cut bees out of somewhere they shouldn't be, like a possum box. And I just find that really rewarding. I get to meet interesting people in the community. I move the bees into a hive, and then I get to manage that population of bees. And it's just something really different that I enjoy doing.